I love smiles. Smiles have a way of bridging gaps, sparking joy, and sometimes even causing a bit of trouble. I remember as a kid, my dentist said I had a great smile. Well, until I bit him. Back then, I might have not appreciated my pearly whites, but it turns out there's a complex world of biology behind every toothy grin. MSX1 or MSH homeobox1 might not be a household name, but it's a master designer in the world of genetics. This remarkable protein plays a critical role in forming our teeth and shaping our face, orchestrating the complex intercellular mechanisms at play to craft our smile. MSX1 is a member of the muscle segment homeobox gene family. The three-dimensional structure of mouse MSX1's binding domain is depicted on screen, featuring three distinct alpha helices. These helices are primarily held together by hydrophobic interactions and are further stabilized by lysine and glutamic acid salt bridges that link the secondary structure into the tertiary structure. This domain consists of 60 amino acids in a highly conserved structural motif known as a homeobox binding domain. Homeobox binding domains are critical for gene expression in embryonic development, interacting specifically with TAAT binding sequences on the major groove of target DNA, regulating gene expression. Well, MSX1's binding domain has been discovered for years, and despite recent in silico modeling of this protein, the exact three-dimensional structure of MSX1 remains elusive. Here's my lousy attempt at predicting the structure using Google's CollabFold. You can obviously see that there's something else at play, like perhaps an unaccounted for post-translational modification, or previously undocumented protein substructure. However, despite the limited understanding of MSX1's structure, its functions are numerous and well documented. For example, MSX1 interacts heavily with the bone morphogenetic protein, or BMP signaling pathway, a critical pathway in the development of dental tissue, influencing cell differentiation and proliferation in dental epithelium and mesenchyme. The study depicted on screen investigates the interactions between MSX1 and Bmp-4, a key protein in the BMP signaling pathway. It documents a positive feedback loop between the two proteins that plays a pivotal role in delineating the areas for tooth germ formation. The interactions of these molecules orchestrates the correct environment necessary during various stages of tooth development. Thus, it's no surprise that mutations in this protein cause Witkop disease and Wolff-Hirschhorn syndrome, which both lead to missing or abnormal tooth and nail development. It is also associated with cleft lip and palate, which cause facial deformities around the nose and mouth. MSX1 is a fascinating protein that plays a critical role in our development. So the next time you flash a smile, remember MSX1, a tiny but mighty architect of our grin. I'll link my sources up here on screen and also down in the description. Thanks for your time and consideration.